Hello and welcome to the NIJ Forensic Science Research and Development webinar series. Today we will hear from Dr. Donald Siegel, Principal Scientist at the New York City Office of Chief Medical Examiner, on validation of a confirmatory proteomic mass spectrometry body fluid assay. Without further delay, I will turn things over to Dr. Siegel.
Hi, my name is Don Siegel. I'm with the New York City Office of Chief Medical Examiner, where I lead the Emerging Technologies Group. The name of my talk today is Validation of a Confirmatory Forensic Body Fluid Assay using Proteomic Mass Spectrometry. One of the most frequent questions I get when I present at meetings is, why do we still need serology in an era of DNA identification? DNA can identify an individual, but serology can give a context to a case. This is especially true with increased sensitivity of DNA testing, as an accused may justifiably claim that their DNA profile is a result of touch, normal epithelial cell shedding, or secondary transfer. Serology can identify the source of DNA in a stain or in an object, for example, blood, saliva, or semen. Defining the problem, current forensic testing and its limitations. Most forensic serology testing being done today is presumptive. That is, they can suggest the substance may be present, but they lack certainty. These tests are usually chemical, enzymatic, or immunochromatographic. Blood tests, for example, are typically the Kastle-Meyer, which is a catalytic reaction with heme, and it's presumptive. False positives include vegetable peroxidases, horseradish, broccoli, etc. Saliva, an enzymatic assay, looks at the enzyme activity of amylase, which is present in saliva. It's also presumptive. Their false positives can be the result of plants or bacterial, which also have amylases. Amylases are also present in other body fluids, and enzyme activity declines with the age of the sample, meaning that after a couple of days or weeks, the enzyme loses its activity. Semen testing is also presumptive. The most commonly used method for identifying semen are the immunochromatographic tests, such as the image you see on your right with PSA, which is a Certec card, or ELISA testing in the 96-well plate to its right. And antibodies are not confirmatory, so they can identify things that they were not generated against, and antigens can be found in non-targeted tissues. For example, PSA can be elevated in female breast cancer patients. The only confirmatory test and the confirmatory tests verify the presence of a specific molecule or compound. That's the difference between confirmatory and presumptive tests. The only currently used confirmatory test frequently used in forensics lab is for semen, and that's looking at semen under a microscope. The problem with that is it's time consuming, and some men don't produce sperm. Why use proteins for body fluid identification? To give you a quick review of what proteins are. Proteins, like DNA, are long, unbranched linear molecules. They're composed of 20 different amino acids in a specific order for each protein. That makes each protein sequence unique. Proteins typically range in size from several dozen to thousands of amino acids in length. Proteins are more stable than nucleic acids in general. So why use proteins for body fluid identification? This is the cover of the May 29th, 2014, volume 509, and the cover is Human Proteome. And the articles were draft map of the human proteome and mass spectrometry-based draft of the human proteome. So the human proteome has been mapped since before 2014, and in fact, since 2014, the depth of the mapping is been greater. The mapping of the proteome is similar to the original Genome Project. This is a figure from one of those papers, and it shows that each organ can be identified by its specific proteome. So if you look at the figure on the left, you can identify brain, kidney, liver, gallbladder, colon, just by looking at the proteins that are expressed in those tissues. The same thing with different body fluids. In fact, if you look at the figure on the upper right, you can distinguish a fetal liver from an adult liver, or a fetal brain from an adult brain, just looking at the proteome. So this basically demonstrates that protein expression can be used to identify tissues and body fluids. So why use proteins for body fluid identification instead of nucleic acids? Protein specificity. Proteins have specific functions in each body fluid. For example, hemoglobin in blood carries oxygen, amylase in saliva contributes to digestion, and semen in semen is part of reproduction. So they have specific functions in each body fluid. They're also present at very high levels because of their specific function. So for example, semen in semen is present in large concentrations, and you don't find semen in blood because there's no function for semen in blood. So because these proteins are present in large amounts, they give you very good sensitivity. Analysis of proteins for body fluid identification also allows simultaneous detection of mixed body fluids. And like nucleic acid, proteins can be sequenced, so they're confirmatory. They're, as I mentioned, very stable. They're less labile than nucleic acids. They give you automatic species identification, and they're amenable to high throughput assays. So why mass spectrometry to evaluate proteins? Mass spectrometry effectively sequences proteins. Consequently, it's confirmatory. Mass spectrometry is accurate, sensitive, and rapid. And mass spectrometry is unbiased. You don't tell the mass spec what body fluid you believe is present. It tells you what body fluid is found in the sample once it's presented. It's run through the LC-MS. So what you see below is the HPLC that we use, the high performance liquid chromatography instrument we use in our lab, as well as the mass spec that we use, which is a SciEx triple core. So just a quick review of how proteomic mass spectrometry works. Basically, a mass spectrometer is a scale, and it can measure proteins extremely accurately to the sub-Dalton level. As you all know, Dalton is the mass of a hydrogen atom, so the accuracy of mass spectrometry is quite good. And it is based on mass that you can distinguish proteins. This is just a simple diagram of what a mass spectrometer is. It basically has three components. So on the left is the HPLC, and the HPLC then submits the sample to the mass spec. So the three components of a mass spec are the ion source, which is where proteins are ionized, the mass analyzer, where proteins are separated, and then a detector. So protein preparation for mass spectrometry. Full-length proteins are too large for most mass spectrometers to analyze. Consequently, proteins are digested to smaller peptides, and the peptides are what is analyzed, similar to what we do with DNA, with shorter segments of DNA being sequenced, and then you can, if you want, you can concatenate the amplicons and find out what the full sequence is. So to break proteins into smaller parts, we use trypsin. Trypsin digests proteins typically into fragments that are between 600 and 2,500 Daltons. The image on the bottom is just an example of a protein being digested by trypsin. Trypsin cleaves at specific amino acids, arginine and lysine, and those, after trypsin digestion is complete, those peptides are available for analysis by mass spectrometry. This is just to give you an idea of what mass spec data looks like. So here, for example, is data from our instrument, and you can see human hemoglobin peptide with a mass of 1378.74 is easily distinguished from a human hemoglobin peptide with a mass of 1314.710. So the criteria that we used for marker selection were specificity, sensitivity, and confidence. Specificity, of course, was the most important. So here at the OCME, we established our own proteomes for blood, saliva, and semen, and compared them to 12 other body fluid proteomes that are already available in the literature to identify those proteins that are highly enriched in each body fluid. This allowed us to exclude proteins, for example, like albumin, which we find in almost all body fluids, and focus on proteins that are highly enriched in the body fluid that we're interested in. Once we identified those proteins that are highly enriched, we selected those that are most abundant to look at as potential markers. The reason for that is the more abundant a protein is in a sample, the greater the sensitivity. Finally, for confidence, we selected multiple marker proteins from each body fluid. The proteins we identified that were useful as markers were hemoglobin alpha, hemoglobin beta, BAN3 anion transport protein, which is a protein in the cell membrane of erythrocytes. Erythrocytes are the most abundant cells in blood. For saliva, we identified alpha amylase, cystatin SA, protein leg 1 homolog, and histatin. And for semen, we identified seminogellin 1, 2, and prostate-specific antigen. So the marker proteins were identified, but to increase confidence that we actually identified the protein that we're looking for, we decided to digest those proteins and look for, when possible, 3 peptides from every protein. So every protein is identified based on the detection of 3 peptides from that protein. So here you see on the right an image of seminogellin 1 and 3 peptides that we chose to identify seminogellin 1. On the bottom of the screen, you'll see the sequence of seminogellin 1 and the location of those peptides within the protein. So now we had multiple peptides from each protein, and typically we chose 3. If you look at saliva, histatin 1 and leg 1 homolog protein are very small proteins, and only 1 peptide was able to be identified from each of those proteins. Are multiple peptides per protein enough? No. It is possible, especially at a crime scene, that somewhere in nature a peptide has the same mass but different amino acid sequence as one of our body fluid markers. So what you see is a cartoon of a mass spectrometer with the ionization, the 3 parts of the mass spectrometer: ionization, Q1, which is the mass filter, and the detector. So sample is ionized before it enters the mass spec. The Q1 is a filter that we can set to allow only a specific mass to exit to the detector. Here you can see that hemoglobin subunit peptide of 932.09 Daltons was selected. But it's conceivable that in nature there is another peptide with 392.09 Dalton mass. In that case, we would assume that the peptide that came through was hemoglobin when actually it could be something else. What I've shown on the right is the peptide sequence of the actual hemoglobin beta subunit peptide, and below that an unknown peptide, which is basically just the same amino acids but reversed in direction. Both would have the same mass. So how do we distinguish that? We use tandem mass spectrometry. So in tandem mass spectrometry, you have ionization, and the first quadrupole is a mass filter that selects a specific mass to enter the second quadrupole, which is used as a collision chamber. So in the collision chamber, the mass of the peptide is fragmented because it interacts with nitrogen molecules, and those fragments can then be detected. So if you look at Q3, mass filter again, we can select specific fragment ions of the original parent ion, and those fragment ions can differ from each other. So if you look at, for example, they're also called transitions, fragment ions. So transition 4 has a mass of 389.45, and if you add a leucine amino acid to it, the mass increases to 502.16, and so on and so forth. So when you have all these masses and you add them together, the sequence of the peptide can only be what you see on the right. This is just an example of MSMS, where we've sent through, the top image is mass spectrometry, which I showed you earlier. So you see the masses of all the peptides, and then you select a specific mass, in this case 1314.710, and you send it through the mass spectrometer again, which is milliseconds, and it's fragmented again, and there you see the fragments of the peptide. You look at those fragments and say, okay, these fragments could only have come from a specific sequence, which in this case begins with VNB, which is also from hemoglobin. So basically, the way the assay works is that you have a peptide, a protein in the upper left-hand corner. Here it's semen and gelin with 3 peptides that we select. So now we have one marker, protein, 3 peptides from that protein, so 3 peptide markers. Each peptide enters the mass spec for MSMS. So first it's selected in Q1. We only allow a certain peptide in. In Q2, there's the collision chamber where the peptide interacts with nitrogen and fragments, and Q3, we detect the fragment ions. On the right, you'll see what the data actually looks like, and I'll get into this in a minute. But basically, those fragment ions all come out at the same retention time, which is here, and we can measure their intensity, which is shown here. So, what does the OCME mass spec molecular serology assay analyze? We look for 29 peptides from 3 body fluids and 116 transitions from all peptides. These are analyzed in every run. So the method that we're using is called targeted analysis with multiple reaction monitoring. We employ this with the triple quad that I showed you earlier. So the multiple peptides are identified and targeted in each sample. So there are 29 peptides for saliva, semen, and blood, and every marker peptide is fragmented by MSMS. So we're looking at a total of 116 fragment ions per run, which takes about 20 minutes. The samples we validated were neat, blood, saliva, and semen, mock samples from the same body fluids, age samples of the same body fluids, mixed samples, 2 and 3 body fluid mixtures, non-targeted samples. So for example, vaginal fluid, animal blood, animal saliva, and animal semen, because these should not give a positive result. We looked at degraded samples of the same 3 body fluids, and what were the performance that we validated for the assay? Accuracy, sensitivity, false negative rate, specificity, false positive rate, positive and negative, predictive values, and repeatability. There was a total of Actually, this number is larger now. There was a total of 2,600 samples. For the protein quantitation assay, there was 1,476, and for the mass spectrometry classification assay, there was 1,124. As I mentioned earlier, these are the criteria that we use to identify that we do have the correct peptide. So a peptide fragment ion analysis includes retention time. All the fragment ions should come out the same retention time, the peak area, and the fragment ion score, which I'll get to in a second. So retention time, because they're all fragmented, all the peptide comes out at one time. All the fragment ions should also come out at one time. So here, for example, is an example of our data for this hemoglobin peptide, and you can see that all 4 transitions are coming out at the same time. Peak area. We measure the area under the curve of each of the fragment ions. And these differences are consistent between runs. So the peak area of fragment ion Y3 on the bottom is consistent throughout the different number of runs as our fragment ion Y7, and this has been shown throughout the validation that these peak areas are consistent, and they have to fall within the expected range. Finally, the fragment ion score. The fragment ion score compares the experimental peak data to the expected pattern using a normalized contrast angle. The result of the score is between 0 and 1, and the closer the scores to 1, the closer sample peaks areas are to the expected values. So the ratio, basically, of the peaks is always the same, because the peak areas are always the same. So what constitutes a positive body fluid identification? These are the markers, as I've shown you before, for blood, saliva, and semen. There are 9 for blood, 8 for saliva, and 9 for semen. Currently, for a sample to be detected, all 9 markers must be identified for blood. Inconclusive, at least 4 but less than 9 markers are identified. And if we detect 3 or fewer, we classify the sample as not detected. Similarly, for semen, 7 out of 9 semen markers need to be identified, including at least 2 peptides each from semen-Magellan 1 and semen-Magellan 2, and all KLK3, which is also known as prostate-specific antigen peptides, must be identified. Inconclusive, again, it is at least 4 peptides, but does not meet the criteria for detected, and not detected is 3 or fewer peptides. Saliva, we have to identify 7 of 8 saliva markers, including all 3 peptides from cystatin, amylase, and either cystatin 1 or leg 1. Inconclusive is the same, similar for the other 2 body fluids, at least 4 marker peptides and at least 1 from amylase, but does not meet the criteria for detected, and not detected is 3 or less or fewer peptides. This assay has been run with samples from vasectomized males. 7 samples were extracted. All gave positive results, so vasectomies do not affect detection of semen. Limits of detection are shown here. So for blood, if we identify 100% of the markers, that's equivalent to about 8 nanoliters of blood. 100% of the markers for saliva is equivalent to about 1.3 microliters of saliva. And for semen, 100% of the markers can be detected in 4 nanoliters of semen. Non-human samples, we ran blood, saliva, and semen from a variety of animals, and as you can see, none of them gave positive results for any of the body fluids. Gorilla was close because it's a primate, evolutionarily close to humans, and we identified 8 peptides that are shared. but one peptide that is not. Confidence in a positive result. So this shows our precision and false discovery rates, and as you can see, we've had no false positives, and so far we have also had no false negatives. The assay has been accredited by ANAB and the New York State Commission on Forensic Science. And I would like to thank the following people and NIJ for their support. Yi Yang runs the mass spec instruments in our lab. Aaron Butler is our informatician. Iman and Tatiana and Jahira all run samples. Tatiana is actually lab manager. Samantha Manure and Sabrina were students in the lab when we did the developmental assay. And David Fenyo is Director of Systems Computational Biomedicine at NYU, who helped with the statistics. And of course, this could not have been done without the support of the National Institute of Justice. And just should point out that opinions expressed in this presentation do not represent the official position or policies of the U.S. Department of Justice or the New York City Office of Chief Medical Examiner.
Yes, thank you so much, Dr. Siegel, for your presentation. I am John Grassel, a Forensic Science Program Manager at RTI International. It was a fabulous presentation. And Dr. Siegel, we have several questions from the audience. And if there are additional questions, please, everyone type them in. And if time permitting, Dr. Siegel can answer them. So our first question, Doctor, is, are there any purification steps after samples are extracted from evidence?
So that's a good question. Actually, no. The samples are extracted from typically clothing, buckle, swabs, and after extraction, the samples are processed immediately for the proteins. And the reason for this is that we don't want to eliminate anything that might be there, and we don't want to enrich by any purification method. We don't want to enrich certain proteins over others. So we keep the sample as it is.
Great. Thank you. Our next question, how was the number of required detective markers determined for a detected, inconclusive, or not detected result?
Yeah, that's a good question. So from the beginning, we decided that looking at all the data that we ran, the frequency at which we found each peptide, but the prime directive of the whole assay was to have multiple peptides and proteins for each body fluid. So that's why we selected 9 when possible for blood and semen and 8 for saliva. And looking at the data from known samples in serial dilutions, we could detect most frequently. For blood, it was very easy. For semen, I think there was only one peptide that we might have lost on a serial dilution, but there was no evidence that, and it was always consistent peptide. There was no evidence that something else was there. Looking at all samples, all body fluid types. With respect to inconclusive, if something is severely diluted, or if there's a mixture where we find one body fluid with all peptides, but another with only partial, we had to make a decision. Are the number of peptides detected sufficient for us to be confident that what we see there is accurate? So that's how the numbers were determined.
Thank you. Our next 2 questions, Doctor, deal with extraction. So the first was, what extraction method did you use?
So we're currently using a bile salt to sodium deoxycholate. And the reason that was chosen was we looked at other detergents, bile salts, chaotropic salts, and none of them, if you use detergents such as SDS, which is an excellent detergent to extract either DNA or protein, you have downstream issues, both with respect to cleaning it up for the mass spec, which doesn't like SDS, and also with respect to interfering with mass spec results. The difference between the two was not that great. Sodium deoxycholate was easy to get rid of, and so it was mass spec friendly. And because the difference wasn't that great, we decided to choose sodium deoxycholate, which is basically bile salt in water. The extraction method is relatively simple. We just take the piece of evidence, we put it with sodium deoxycholate in a small volume, we vortex it and let it set for about 30 minutes, and then the sample is considered extracted, and we use the extract after spinning it down to do further processing, which means basically just trips and digestion, and then it goes on the LCMS.
Great. And I know you said you use just a small amount for extract. One of the questions was, how much extract do you need for the analysis?
So when we were developing the assay, we developed it so that we would use the smallest amount possible. So we take typically a piece of evidence that's no bigger than 5 by 5 millimeters, and we use 200 microliters of sample to extract it, and we have good results from that small volume.
Which brings us to our next question, which is always on the minds of lab managers. How much does the assay cost?
Yeah, I get that question all the time. So the good news is the assay itself is cheap because the reagents are cheap. There are no kits. You just need trypsin. That's the most expensive reagent that we use. So the per sample cost is, I think, the last time I calculated, about $1. I think that included the plasticware. Of course, the mass spec costs money. Many labs have mass specs in their toxicology department. So that's, if you have time that you, from toxicology, where it's there, you can share a mass spec, that's one way to go. But an LC mass spec is expensive. It's probably 4 or 5 times more than well, maybe 3 or 4 times more than a CE instrument. So with respect to DNA, I would guess we're talking about $500,000 for a mass spec.
Well, thank you. So lesson learned: be nice to your toxicologist in your lab. Maybe they'll
Right.
let you use it. Our next question is, how many of the non-human samples were inconclusive?
I'd have to go back and look. I don't think any of the non-primates were. I think they were inconclusive. I think the only non-human sample that was inconclusive was gorilla, if I remember correctly. But I'd have to go back and look to confirm that.
And speaking of samples, have you tried it on old or degraded samples?
So we've made mock samples over 10 years now. We don't put them on the roof. We just make the samples. We leave them in a hood in the lab, and every so often we go back and test them. So for degraded samples, we've never not gotten a positive result. Although after 10 years, the signal might be down, but it's still sufficiently high to be conclusive what it is. We've also taken PT samples from the lab that are used for DNA analysis that have been more than 10 years old, that were left over, that weren't used, they weren't needed, and we've gone back and extracted those, and we've always gotten a positive result.
Sort of in line with that, What do we know about the material donors? Was everyone healthy, and were any interference? Have any interference been identified?
So everyone was healthy. We've gotten hundreds of samples. NIJ was very gracious. We've also looked at menstrual blood and vaginal secretions. So everyone was healthy. There was no person with a known disability. I wouldn't expect problems with blood because problems that you would see with blood that would, that people weren't healthy would probably be to the point where it would be pathogenic to them. Semen, we've only looked at normal volunteers and vasectomized semen. We are currently establishing a database for polymorphisms in the peptides that we're looking at, so we can add those if their population frequency is high enough. We will add those in the future to the assay.
Great. And did you find that the DNA extraction result was the same whether it used the manual protocol or the kit-based method?
So we did do some. So the assay currently is the way it works here at the OCME. They do a cutting for us, and they do a cutting of the sample for DNA at the same time. We have demonstrated, and again through the generosity of NIJ, that we can do a simultaneous DNA protein extraction, and we're looking into introducing that in the future. There were no problems. There is a problem. The kits are designed for so those are kits. So they're designed for that. You can't use current methods of DNA extraction that use proteinase K because you digest proteins into amino acids. But the kits that are designed for DNA, RNA, and protein simultaneous extraction use trypsin instead of proteinase K. And we didn't see a decline in STR profiles.
How long would you say it takes from the time you receive a sample until you report out the results?
Yeah, so that's critical for us. The assay itself, if you got a sample on Monday, you could have a result on Tuesday. The problem is, as everyone is probably aware of, when you're in a forensics lab, there are certain protocols that have to be followed. LIMS is, you have to enter everything into LIMS. So we currently, it probably takes, if there was a rushed sample, we could probably have it in the next day, but most likely 2 or 3 days to run. But that's not just one sample, so we do them in batches. So whether we did 50 or we did 1, it would take the same amount of time. The longest part of the assay is the overnight digestion, because the extraction is 30 minutes and the LCMS is 20 minutes. It's the overnight digestion with trypsin, which we're looking into. There are products available that can apparently do it in 2 hours, and we're investigating those to make it shorter.
Is it possible to detect and identify a mixed sample, such as blood mixed with semen?
Yeah, so that was part of our validation. So we've done mixtures of 2 body fluids, and we've done mixtures of 3 body fluids. I should just preface this by saying we're looking at proteins, and the protein content, that's why the volumes that we can detect of blood, saliva, and semen are different, because it depends on the amount of protein in a particular body fluid. So blood, for example, has about 300 micrograms per ml, semen about 100, and saliva about 10. So we have been able to detect mixtures when the amount of protein present was, enough to detect them. So yes, we've gotten full profiles, and in some casework samples, we've gotten full profiles as well as some partial profiles. So one body fluid is a full profile. The other body fluid might come up as a partial profile, depending on what the ratio is.
And a couple of questions, both related to future studies. So one being, do you have plans for future study on this topic? And I know you alluded to that. And then the other follow-up question is testing of the effects of environmental factors.
Right. Somebody else had asked a question about inhibitors. So far, and I neglected to include that in my previous answer. So far, we haven't seen anything that inhibits trypsin. We haven't found anything from the mock samples that we've created that inhibits trypsin. Future studies, there's a lot. We have menstrual blood and vaginal fluid that we are currently in developmental validation that we hope to eventually introduce into the assay. As I mentioned, we're looking to reduce the assay time from overnight digestion to a 2-hour or less digestion. We've also are looking to add other markers to the assay so we can make it more sensitive. So there's a lot on the plate that we're looking into doing. Yeah.
There's definitely so we have had a lot of questions, but I wanted to take time to ask Dr. Siegel if you had any final thoughts on this subject before we wrap up.
So I think yeah. So I think identification of body fluids has become more and more critical because when you go to court and you have a presumptive test for semen, especially in a sexual assault case, it's not it doesn't bring the same statistical heft that DNA does, and this assay was designed to compete with scientifically and statistically with the quality of DNA assays. And so I think we've done that, and I think that it would the cost of reagents and kits will become available. I think it will become it could very easily become the body fluid test to go to. It is confirmatory, and it's something that's really needed.
Thank you again. And if there are no further questions, we're going to go ahead and wrap up today's webinar. Please feel free to contact our presenter, Dr. Siegel, if you have any further questions. Thank you so much for attending, and thank you, Dr. Siegel, for being here with us today.
It's my pleasure. Thank you.
As you depart, we kindly ask that you take a minute or two to complete our post-event questionnaire. The link will be pasted in the chat and also appear as you exit the Zoom room. Your feedback helps us improve our outreach efforts and continue to provide the best experience possible. Thank you again for attending, and we hope everyone has a great day.